It has been estimated that humans consume 5 grams of microplastic per week through what we eat and drink. That is as much as a credit card. Where do these tiny plastics come from? Most micro and nanoplastics come from the weathering of larger plastics that we throw away. They are a pollutant found in every imaginable ecosystem. So what are they doing to our bodies? Ingestion of microplastics have been shown to induce chronic inflammation, oxidative stress, neurotoxicity, energy and metabolism dysfunction, pulmonary and autoimmune disorders, even loss of bone density. This study by Peterson and Baker set out to test the endocrine disrupting potential of these plastics during development in the zebrafish model. The zebrafish is used in toxicology research because they are small, have a short lifespan, and are transparent for a period of time after birth. Perhaps most importantly, 80% of the zebrafish genes and proteins are conserved in mammals. The team exposed zebrafish embryos to fluorescently tagged polystyrene particles, either 50 or 200 nanometers in diameter, at a range of concentrations over a 5-day period of time. The concentrations were either 10, 100, 1,000, or 10,000 parts per billion. They used the plastic polystyrene because it is one of the 3 most produced plastics globally. Microscopy was used to determine if the plastics accumulated in the body of a zebrafish. This is shown in the picture on the top right. Notice accumulation in the GI tract, liver, and cranial regions. A behavioral swim assay was used to assess changes in their activity during light and dark periods. Exposure to the 200 nanometer plastic resulted in hyperactivity during dark periods at the 2 highest concentrations. Peterson used transcriptomics on several tissues to determine which genes were turned on or off by the plastic exposure. This data was then put into pathway analysis in order to understand the biological significance of these differentially expressed genes. They found that the 200 nanometer plastics at 100 and 1,000 parts per billion cause more than 750 genes to be differentially expressed. This corresponded to several important developmental pathways, including nervous system, neuromuscular function, metabolic, cardiac, hepatic, and epigenetic pathways. The neurobehavioral findings of hyperactivity corresponded with transcriptomic findings in 100 and 1,000 parts per billion exposure. Taken together, the researchers concluded that nanoplastic exposure causes neuromotor impairment and significantly alters normal transcription events. Future studies should use similar methodology to determine the toxicology of microplastics when they become vectors for other molecules in the environment, like pesticides, persistent organic pollutants, and antibiotic-resistant bacteria. Thank you.